Okay. Okay. This really is going to be the last video of this unit, Unit 5, as I promised, and is the video where I'm going to introduce you to the serotonin 5-HT2A receptor. However, first of all, I want to have a quick word about the difference between neurotransmitters, the term you'll probably know, and neuromodulators, which are also neurotransmitters, but I think it's important to make the distinction. So this will most easily be done at the board. Okay, so we have, which neurotransmitters have we looked at? Well, first of all, let's draw a synapse. Here's a synapse, like this. So we have looked at glutamate, for example, and we know that glutamate is released from synaptic vesicles. Here are the vesicles, and glutamate is released, diffuses across the synaptic cleft, and I will draw in some little glutamate receptors. This will do. There we go. Yeah. So we've got something like AMPAs, right? You would know what these are now, the AMPA receptors. Bad handwriting as usual, but whatever. So glutamate is released. So this is glutamate. And we would call glutamate in this instance a neurotransmitter, because it's transmitting the information across the chemical synapse, right? This is the chemical synapse, and information is transmitted. And it is a neurotransmitter. However, serotonin, or 5-HT, as well as dopamine and some others, but anyway, we'll stick with those, are more what we would call neuromodulators. Do I need to write that out? I'm going to write it out anyway. I'll write it at the bottom. Neuromodulators. Okay, in fact, I think I might even clean off this board. So neuromodulators, whereas neurotransmitters tend to have a very fast and short-lived effect. Glutamate is released from the presynapse, has a very rapid action, and we saw the effect on the membrane potential, right? We're at resting potential. We get this little EPSP caused by glutamate, which is very, very short-lived. And that's really important because you don't want this effect lasting too long, otherwise it's very difficult to reliably transmit information if these effects last too long. Now, what's notable about this synapse is that the distance here is very, very small. The distance, the width of the synaptic cleft is kind of narrow, right? And that's so that glutamate doesn't kind of spill out of the synapse. You want it to be a tight one-to-one connection between the presynapse and the postsynapse. And sometimes I like to refer to this, as do others, as wiring transmission, because it's the basis for the wiring of the brain, right? These synapses form the connectivity of the brain. So the what's the word I'm looking for? Fidelity, maybe. Kind of the fidelity of these connections relies upon the fact that your glutamate isn't leaking out of the synapse. So the synapse has to be very, very narrow and tightly controlled. Otherwise, glutamate is going to leak out and perhaps affect other synapses. Okay, so that's neurotransmitters. So neuromodulators, slightly different. I may have confused you a bit there, but these are neurotransmitters. So neuromodulators, let's clear this off. So neuromodulators, I'm going to write this again because, if I can. Neuromodulators, as I said, and serotonin 5-HT is an example of a neuromodulator. So neuromodulators, they work in almost exactly the same way. So you've got a presynaptic terminal, you've got synaptic vesicles that contains the neuromodulator. You know, there could be 5-HT in these vesicles, and they release it. However, what you normally find is that the synaptic cleft is much wider. And what that means is that serotonin can actually spill out and can actually start to affect not just one postsynaptic terminal, but can actually start to affect others, like this, and can affect neurons more generally. So neuromodulators, rather than being important for kind of the wiring of the brain, are more used to modulate, yes, that's where the term comes from, to modulate or modify the activity, the behaviour of neurons on a slightly more long-term scale. So serotonin, when serotonin is released, it spreads out of these synapses and can actually kind of bathe large numbers of neurons and have effects on large numbers of neurons at the same time. And that kind of changes the overall behavior of lots of neurons, perhaps over a much longer period of time, certainly a lot longer than the millisecond or so that you get with neurotransmitters. So whilst serotonin is technically a neurotransmitter, it fulfills all of the requirements to be defined as a neurotransmitter. It's useful to make that distinction between a neurotransmitter and a neuromodulator because serotonin is a neuromodulator. Okay, I think that's enough there. So, as promised, we will now discuss the 5-HT2A receptor. And this is a serotonin receptor, of course, and it's one of a number of serotonin receptors. We're not going to look at the other serotonin receptors. Of course, they all bind to serotonin. Serotonin is the ligand for all of these other types of serotonin receptors. Why are there many types? Well, they all have different things. They all have different functions. That means they are structurally different. They interact with different signaling pathways inside the cell. They have different effects on the cell. So serotonin has, as a result, serotonin has many effects depending on, the effect of serotonin on a neuron will depend upon which serotonin receptors are on that neuron. If a neuron has, 5-HT2A and 5-HT1A, maybe 5-HT2B, then the overall effect of serotonin will depend upon the interactions with those receptors. So you can't kind of ascribe a particular function to serotonin and say, oh, serotonin does this. Serotonin doesn't really do anything apart from interact with serotonin receptors. The actual effect will depend upon which serotonin receptors are actually embedded in the neuronal membrane, and that will vary depending upon which type of neuron it is, where it is in the brain, and things like this. It's complicated. It's complex. Okay, so first of all, let's just talk a little bit about serotonin receptors. So there are a number of serotonin receptors. They are all, if you read them in the biochemical literature, you will see all of them prefixed by 5-HT, and that stands for 5-hydroxytryptamine. Yes, it is a tryptamine, just like dimethyltryptamine or 5-methoxydimethyltryptamine or 4-phosphoryloxydimethyltryptamine, i.e. psilocybin. And we will look in the next unit, early in the next unit, we'll look in more detail at the structure of these and how they're related. But that's where it comes from. So that is serotonin. And the 5-HT receptors, which you'll often see abbreviated just as 5-HTR, fall into a number of subtypes. So you have 5-HT1 receptors, 5-HT2 receptors, all the way to 5-HT7 receptors. Now, some of these are also broken up into further subtypes, which are normally given a letter. So we're only interested really in the 5-HT2 receptor, which has 3 subtypes: the 5-HT2A, the 5-HT2B, of course. This is not rocket science, it is neuroscience, and the 5-HT2C. And this is the baby that we're all interested in. Okay, so it has been established now for a number of years, many years really, through many lines of evidence, including the use of antagonists at the 5-HT2A receptor. So if you give anyone, an antagonist at the 5-HT2A receptor, there's a very important antagonist called ketanserin, and then you give them a psychedelic drug, they won't experience the effects. And this has been done with psilocybin specifically, and you can look this up online if you're interested in that. And there are other lines of evidence as well, and it's still something of a very active area of research in understanding what it is about the 5-HT2A receptor that gives it its kind of psychedelic effects, or why activation of the 5-HT2A receptor generates psychedelic effects in humans. And this is something that we're going to cover. So you will understand all about this by the end of the next unit, maybe, or maybe the unit after. Certainly by the end of the course, of course. But in this video, all I want to do is talk you through how the 5-HT2A receptor works. In other words, I'm interested in telling you about the signaling pathway. Okay, so let's bring up the signaling pathway for the 5-HT2A receptor, and we will go through it carefully. Bear in mind, make sure that you've watched the rest of the unit up to this point, because otherwise some of these ideas will be a little bit confusing for you if you're not familiar with intracellular signaling pathways. Okay, so let's have a look. Okay, so this is a simplified signaling pathway for the 5-HT2A receptor. So this in red here is obviously 5-HT, this is serotonin. And this is the 5-HT2A. Put the R there, the receptor. So we'll go through this kind of step by step. So 5-HT binds to the 5-HT2A receptor, of course. You get this change in conformation, and it binds to a specific protein that you might have met before if you studied biochemistry, but otherwise, here it is. And this is called GQ. Now, this GQ falls into a family of proteins called, wait for this, G proteins. And they're really important because they are often the kind of the initiator of the intracellular signaling pathway. They provide the connection between the receptor and a particular intracellular signaling pathway. So they're very important, and we are going to meet them again in the next unit. Why it's called GQ doesn't matter. Don't worry about that. But just when I say GQ, you know what I'm talking about. I'm talking about a G protein that interacts with the 5-HT2A receptor when it is active. Okay, so GQ, when it binds to the activated 5-HT2A receptor, activates a protein called PLC. Now, PLC stands for phospholipase C. Again, the name isn't important here. The important point is that it's activated by this GQ protein. And you know now what I mean by activating. The active site is opened up. Now, phospholipase C, or PLC, is an enzyme that breaks down this molecule called PIP2. So PIP2 is the substrate for phospholipase C, and basically it's phospholipase C splits PIP2 into two smaller molecules. One is called DAG, which stands for diacylglycerol, and the other is called IP3. Again, names aren't important here, but you've got two molecules. Now, these two molecules then have two distinct effects, but which are ultimately connected. So IP3, indirectly and through a rather complex mechanism that I'm not going to go into, causes the level of calcium inside the cell to rise quite dramatically. So it leads to a dramatic increase in the concentration of calcium. Specifically, these are calcium ions, or I should really put Ca2+ here. DAG, on the other hand, is involved in the activation of, it actually binds to a protein called PKC. Now the K here, to those of you who were listening earlier in the unit, should give you a clue as to what this is. It's a kinase. It's in fact protein kinase C. A very, very important protein kinase, and a rather complicated one. Its mechanism of activation is very, very complex. However, I will tell you simply that protein kinase C is activated by both DAG and calcium. In fact, when it's highly activated when there's both DAG and calcium. So together, overall, the activation of phospholipase C causes a dramatic activation of PKC. Now, PKC then, through a number of steps, a number of stages, so a number of kinases, leads to the activation of this kinase called ERK, E-R-K. Sometimes you will see ERK referred to as MAP kinase. Again, names aren't important, but it's a kinase. It's phosphorylated and activated, and the activated form of ERK does what? Well, it phosphorylates a potassium channel. Now, we haven't discussed the phosphorylation of potassium channels, or any channels really, but just like enzymes, sometimes when you phosphorylate a channel, you can actually affect its structure. You can cause its structure to distort, and sometimes that can cause the channel to kind of be more likely to be open, or sometimes it can cause it to distort and kind of block it. So you actually kind of inactivate the channel, and that's what's happening here. So ERK, so let's have a look. So ERK here is actually phosphorylating this potassium channel, and as you can see by the flattened arrow here, it's actually inactivating the potassium channel. So what effect is that going to have? Now we need to think about this. Okay, so activation of the 5-HT2A receptor causes inactivation of these potassium channels. Now, first of all, when I say potassium channels, I don't mean the voltage-gated potassium channels that we met in the first unit, right? These are a special type of potassium channel. There are many types of potassium channels, just like there are many types of sodium channels and other calcium channels, etc., etc. So we're just going to call them potassium channels, but actually it's a specific type of potassium channel. But that's not important. The point is it's a channel that can be open or closed, and that allows potassium to travel through. Now, here's the point. So going back again to the first unit, we discussed ionic gradients across the neuronal membrane. Right? The simple idea that there's a high concentration of sodium ions outside the neuron and a low concentration of sodium ions on the inside of the neuron, whereas it's the opposite for potassium. There's a high concentration of potassium inside the neuron and a low concentration outside. So the concentration gradient for potassium is going upwards, upwards, out of the cell. It's going out of the cell. So we need to think then, what is going to be the effect of kind of inactivating, stopping these channels from working, stopping them doing their job of allowing potassium to flow through. Now, one thing we didn't really discuss in the first unit, we discussed this idea of a resting potential. The idea that the membrane potential sits at a kind of fairly stable, not completely kind of fluctuating level at kind of minus 70 millivolts, right? Now, what we didn't discuss was what actually sets that membrane potential. And it's complicated, and it involves a lot of kind of electrochemistry and stuff that we're just not going to get into. It's not important. But what you need to understand is that there are many types of ion channels, and ion channels, of course, they allow ions to move through, and ions, of course, they carry charge. So when ions move either in or out of the cell through these channels, they're going to affect the membrane potential. They're going to affect the distribution of charges, the charge separation across the membrane, which of course is the membrane potential. So we'll look at this in a second on the board to make it clear. But basically, you've got some channels are going to be kind of pushing the membrane potential upwards. They're going to be have a kind of a depolarizing effect, whereas other channels are going to have a hyperpolarizing effect. For example, if you have some sodium channels in the membrane, not the voltage-gated ones, they're just for the action potential, just some other kind of sodium channels, right? And let's say some of these are open. Well, what's going to happen when they open? Well, sodium's going to go in. What's that going to do to the membrane potential? You should know. It's bringing positive charge into the cell. It's going to depolarize the membrane. Whereas potassium channels, well, they're doing the complete opposite, because potassium wants to go out. So potassium will flow down its concentration gradient out of the cell, and that will have obviously the complete opposite effect of the membrane potential to sodium. So you've got these channels, and they're kind of pulling in opposite directions. Some want to bring charge into the cell and push the membrane potential upwards, whereas some want to take charge out of the cell and push the membrane potential, positive charge, out of the cell and push the membrane potential downwards. They want to hyperpolarize. So you've got this fight, and wherever the fight kind of sits is the resting potential. It's kind of imagining it like tug of war. And you've got a group on one side, a group on the other side, and they're pulling on the rope. And where the middle of the rope eventually sits, when they're kind of both teams are kind of maximally straining, it's going to depend on the relative pull of each side. And it's the same principle with the membrane potential. So let's have a look at this on the board, and hopefully it will be clear. So you remember this diagram now. This is very familiar to you. So you've got 0, and then we've got our resting potential, which we will kind of put tentatively, we'll put it here. So this is our resting potential. Now this is determined, as I said, by this kind of balance of push and pull effects. So sodium channels, so let's draw these in, I don't know, blue, that'll do. As I said, we'll want to push in this direction. So we would say they are depolarizing. Whereas potassium, and you know why that is, because sodium channels let sodium in, and they obviously depolarize the membrane. They make it more positive, push it upwards. Now, potassium channels, which will mean yellow, they do the opposite. So K channels are hyperpolarizing. Why? Well, simple enough. Polarizing, that'll do it. Simply because the potassium moves in the opposite direction to sodium, so it's taking positive charge out of the cell, attempting to make the membrane potential more negative, hyperpolarizing, pushing the membrane potential downwards. So there's always this fight between these channels, and this balance that you're seeing here is the resting potential when all of this is kind of at an equilibrium. So ask yourself this question: what happens when we introduce serotonin binding to a 5-HT2A receptor? Well, I want to color this, I don't know. So what does serotonin do? So 5-HT binds to the 5-HT2A receptor, and we know now what that does. It inhibits the K channels. So it removes part of that pulling effect, this hyperpolarizing effect of the potassium channel. So what's going to happen to the membrane potential? Well, you're going to get an increase in the membrane. It's going to be depolarizing, right? Because now the sodium channels kind of win out a little bit more because you've weakened the effect of potassium channels, or at least a subpopulation of certain types of potassium channels have been inactivated by phosphorylation of ERK. So ERK phosphorylates the potassium channels. It basically distorts the channels. It stops them opening properly, doesn't allow potassium to flow out of the cell. So essentially inactivating those channels, which means that the sodium channels have a stronger effect, depolarizing effect, and so you get this depolarizing effect of by inactivation of these potassium channels. Hope that makes sense. If not, yes, go over it again. It's a really important idea, and it follows entirely from what we discussed right back in the first unit. So it's not too difficult. Pretty simple stuff, I think. Yes. So to summarize, serotonin, when it binds to the 5-HT2A receptor through this signaling pathway that ultimately leads to the inactivation of a certain type of potassium channel, pushes the membrane potential higher, towards zero. It depolarizes the membrane. Now, what effect does that have on the neuron? Well, it pushes it closer, pushes the membrane potential closer to the threshold. That is the key. And in the next unit, I need to stop pointing like that. It's quite rude, isn't it? Rude. Anyway, so in the next unit, we're going to look at all of the consequences of it, because fundamentally, it's this depolarizing effect of 5-HT2A receptor activation that is ultimately responsible for the psychedelic effects, or at least a large proportion of the psychedelic effects of psychedelic drugs such as LSD, psilocybin, and mescaline, and DMT. But there's obviously a lot to kind of unpack. I hate that word. Unpack. We need to unpack all of these effects. But this depolarizing effect is fundamental to all of it. So please make sure you understand why when you activate the 5-HT2A receptor, you depolarize the neuron. And I think we're done. I think we're done for this unit. So I will hopefully see you in Unit 6, which is our first kind of psychedelic drug unit. We'll start looking at the psychedelic drugs, and we'll start thinking about psychedelic drugs binding to the 5-HT2A receptor and lovely stuff like that. Wonderful. Okay, fantastic. I will see you in the next unit. Bye. I'm kidding. However, if you do want to support the production of future courses, or you just want to leave a tip, then there are virtual tip boxes below. There's PayPal, Ko-fi, or Ko-fi, whatever it is, even a Bitcoin address if that is your thing. Minimally, please like and subscribe. It really helps me. And please follow me on Twitter and Instagram if you use them, Alien Insect. And I think that's about it. I hope you're enjoying the course. Thank you.